Hi, it's with great pleasure that I'm going to present our work called A Scalable Multivariate Bayesian Polygenic Model for Heritability-based Principal Component Analysis. This work has been done with Thomas Nichols and Habib Ganjgai. So the first question is, what is the ultimate goal of our work? Well, it is to better understand how the human brain works, its organization, its functions, by assessing the impact of genetic variance on neuroimaging phenotypes. So example of neuroimaging phenotypes can be, for instance, resting functional MRI. Now, it turns out that statisticians have come up with a framework to do this. It's called Genome-wide association studies, so to assess the impact of genetic variance on phenotypes. And also, it is known that combining phenotypes yields better power than considering each phenotype separately, or the so-called univariate approach. So now the question is, how do we actually combine phenotypes? And now it's actually interesting to focus on the notion of heritability. When we have one phenotype, we can always decompose its variance into genetic and environmental variances, and the heritability will be the proportion of that total variance that is due to genetic effects. In the multivariate case, when we have multiple phenotypes, we can decompose this time the phenotypic covariance matrix into a genetic covariance matrix and an environmental covariance matrix. And now it has been actually shown that dimensionality reduction method that uses the estimated genetic or environmental covariance matrices to combine phenotypes with maximal heritability outperforms multiple regression. And so the goal of our work is to perform that dimensionality reduction technique in a scalable manner by estimating the covariance matrices, so genetic and environmental ones, efficiently, even when the number of phenotypes is big or the number of people in the study is big. Let's now focus on the model we're using to estimate those covariance matrices. Say we have n individuals, p phenotypes, and k covariates we're trying to control for, such as weight or sex. Then big Y is the matrix of phenotypes, big X the matrix of covariates. And what we're using is a linear mixed effect model, which is essentially, as you can see, linear regression, except we have two sources of randomness, G and E. Because we're in a multivariate setting, those things are matrices, and G and E follow what is called a matrix-variate normal distribution. So it basically means that each coefficient of those matrices is normally distributed. Now let's look at G, which is the genetic source of randomness. It is controlled by two things, phi, which is the kinship matrix or genetic relatedness matrix, which tells us how the individuals in the study are genetically correlated, and sigma G, which is the genetic covariance matrix that tells us how the p phenotypes are genetically correlated. Then E is the environmental source of randomness, and it is controlled by sigma E, which is the environmental covariance matrix that tells us how the phenotypes are non-genetically correlated. Now, on top of that, we actually build a conjugate hierarchical Bayesian model for the covariance matrices. Now the question is, how do we actually estimate our covariance matrices in a scalable manner? So first, the method we're using is the expectation maximization, or EM, algorithm. Now the step one to make it scalable is to actually simplify the model I was showing before, the linear mixed effect model, by applying an orthogonal transformation based on the eigenvectors of the kinship matrix. So it allows to diagonalize the kinship matrix, and working with diagonal matrices is always easier in terms of calculation. Now the problem remains because, as you saw in the previous slide, we're working with matrix-variate normal distributions, so normal distribution on matrices, and this leads to very heavy calculation. So the solution is to actually define auxiliary variables to work with diagonal matrices, which will allow to perform the updates in the EM algorithm more efficiently. So here, for instance, you see that on the figure we have new variables such as lambda 1, lambda 2 that are diagonal, and this will considerably facilitates the computations. And so we go from an algorithm that is cubic in the number of individuals to linear in the number of individuals. Now that we know how to estimate our covariance matrices, let's focus on how to compute heritability. So in the univariate setting, when you only have one phenotype, you can fit a linear mixed effect model for that phenotype and estimate the genetic variance sigma g square, the environmental variance sigma e square, and then the univariate heritability of the phenotype would just be sigma g square over the total variance as the proportion of the variance that is due to genetic effects. In the multivariate setting, if we're interested in computing the heritability for phenotype I, it is essentially the same formula except the variance for phenotype I is the ith diagonal coefficient of the covariance matrices. And so we have a similar formula. And finally, because that's what's of interest for us, to compute the heritability principal components, we just have to find the eigenvectors of the matrix that is given here that are the directions that actually maximize the heritability. Let's now spend some time on the results. So obviously the first thing we did was to assess the performance of our model on synthetic data, which was a success, and then we moved to real-life data from the Human Connectome Project, HCP, and the UK Biobank, UKB. Now, whether it is the Human Connectome Project or the UK Biobank, our analysis were performed on 4 types of data. So gray matter kinds of data, where the phenotypes are cortical areas and thicknesses, as well as the volumes of the subcortical areas measured with the FreeSurfer software. So those data sets are referred to as FS. White matter data, where phenotypes are related to the integrity of regions in the white matter calculated using the TBSS algorithm, so the TBSS data. And finally, the connectivity matrices, where phenotypes are the pairwise edges of regions of the brains based on their Bol signals. So we have 3 types of data sets of that form, RFMRI 15, 25, and 100. And I really want to point out that, and that's what makes the success of our method. We're able to run our method efficiently on data sets where the number of individuals is pretty large, such as the UK Biobank with roughly 30,000 individuals, and when P is large enough so that we can consider regions all across the brain, for example with RFMRI 100. Let's now take a look at an estimated genetic correlation matrix. So here what you see is the genetic correlations for FS in the UK Biobank. So on the figure, the rows and columns have been permuted to better show clusters of genetically correlated phenotypes. Now if we have access to the rows and column names, which we have but didn't show here for clarity's sake, what this heat map tells us is that there are very strong correlations between same phenotypes that are in different hemispheres, which makes sense. Also high genetic correlation between volumes of the areas of the corpus callosum, and finally, high genetic correlations among the thicknesses of the regions of the cortex. Now our method allows to estimate efficiently the genetic covariance matrix, the environmental covariance matrix, and from these 2 matrices we can build the phenotypic covariance matrix, so the total covariance matrix, by just summing the 2 estimated matrices. And now we can compare that phenotypic covariance matrix to the genetic covariance matrix. So here what we see is the genetic correlations for RFMRI 25 from HCP compared to the phenotypic correlations, so the total correlations for that same data set. And what we see is that we have the same clusters, so the same patterns in the correlation between phenotypes, but the genetic correlations appear stronger. Let's now focus on the heritability of our phenotypes. With our method, we can compute the multivariate heritability of each of the phenotypes using the estimated genetic and environmental covariance matrices as explained in one of the previous slides. We can also compute the univariate heritability of each of those phenotypes, and we don't need our method for this. For instance, we can use the LME-kin package from R to do that. And this is what we did. For each of the phenotypes of the dataset from the Human Connectome Project, we computed the multivariate heritability with our estimates of the covariance matrices and the univariate heritability with the LME-kin package from R. And we computed the difference between those 2 quantities. And this is the figure you see here, the difference in heritabilities for each of the phenotypes, and it shows that those differences are pretty small. And this is great because it means that the heritability, whether it's univariate or multivariate, are comparable. And it's a good thing because if our method was producing something that is very different from the univariate estimate from R, we would have reason to be worried because in the end, we're estimating the same quantity. But this is not the case here. Finally, let's focus on dimensionality reduction as it was one of the goals and compare PCA with heritability PCA, the method where we use the estimated genetic and environmental covariance matrices to extract principal components that have maximal heritability. So let's take a look at the figure on top, the analysis on RFMRI 25 from the Human Connectome Project. Say you're interested in achieving 80% cumulative explained heritability. So this is the green line, roughly 40. Then if you use heritability PCA, you're going to need roughly 50 heritability principal components, that's the red line. Whereas with vanilla PCA, we're going to need more than 3 times that number of components to achieve the same heritability threshold. Same if you look at RFMRI 100 from UK Biobank, the other figure, you see that if you want that 80% a heritability threshold, which is the green line, you're going to need roughly 800 heritability pieces, that's the red line, to achieve the threshold, whereas you're going to need roughly 400 additional components if you use vanilla PCA to achieve the same threshold. So in the end, heritability principal components allows to consider fewer transform traits for a bigger explained heritability. And we would have similar conclusion with FS and TBSS datasets. In conclusion, performing data reduction using the heritability principal components allows to build a smaller number of traits than with vanilla PCA, and those traits will be more likely to detect genetic association, which will result in an increase in power when performing GWAS. Note that we've also developed a stochastic version of our method, where at each iteration, the E-step is calculated using only a subset of the data. That method, the stochastic version, has been tested on synthetic data with success, and one of the next steps would be to actually apply it on the UK Biobank data. One of the future steps would also be to have a neuroscientific interpretation of the heritability principal components we've derived with our method. Thank you.